This video features the usage of Blade in clinical workflow. The medical staff starts by uploading the digital image of the peripheral blood film. The AI model processes this image, and the user can enter the task to review the different white blood cells which have been pre-classified by our AI. Upon finding a misclassified cell, he will simply reclassify them into the right cell type. Next, the user can review the slide image, and he can note any observations in the comments box. The user can also enlist the red blood cell functionality to assist in cell grading. The user selects a region to calculate red blood cells within that region. A reference of the required number of red blood cells for each grade is calculated. As an example, anywhere between 1 to 2 schistocytes qualifies for a grade of 1+. No schistocytes are seen here, hence the grade remains as 0 and no grade needs to be reported. After red blood cell grading, the user can also opt in for platelet estimate. A field of view corresponding to 10 high-power fields is shown, and the user can generate the platelet estimate. Within a few seconds, the average platelet per field and platelet estimate are generated. All of the assessments are collated in a report for final review. Upon completion, the user can sign off the report and have it uploaded into the lab information system.